Dear students, I welcome you all to UGC E-Pathshala. I am Dr. A. Sangamitra, Assistant Professor from Department of Food Technology, Kongu Engineering College. Okay, let me move on to our today's topic on pulsed electric field. Consumer demand for fresh-like food products, mainly liquid foods like fruit and vegetable juices and milk, are increasing due to its richness in antioxidants, vitamins, phenolic components and carotenes. Therefore, it is the sole responsibility of the food manufacturers to retain the original flavor and color characteristics as well as the nutritive value of food during any processing. Traditionally, thermal treatment is carried out to inactivate the microorganisms, but the thermal stress alters the nature of the product. The conventional heat treatment leads to alterations in flavor, taste and nutrients. Hence, the alternative method is the non-thermal techniques. Pulsed electric field is a non-thermal food preservation method for pasteurizing the food products. Pulsed electric field is used in various food processing applications due to numerous advantages than the conventional heat treatment. On completion of this session, you will be able to understand the principle behind pulsed electric field and also to identify the applications and effect of pulsed electric field in food processing. First, we will discuss about pulsed electric field and its principles. Pulsed electric field technology is one of the emerging non-thermal methods of food preservation which is capable of disinfecting food products. It is a promising technique to inactivate microorganisms. Earlier, the electroporation of cell membrane has been used in biotechnological field for infusion of foreign DNA into the cell. In 1925, Betty and Lewis used non-pulsed electric field of 220 volts to pasteurize milk and found reduction in microbial load. A pulse is a single disturbance that moves through the medium from one point to another. Pulsed electric field uses short high voltage pulses to the liquids or semi-solid food placed between two electrodes to preserve the foods. The applied voltage ruptures the microbial cell membrane and inactivates the endogenous enzymes at or near room temperature. Electric fields in the range of 10 to 80 kV per centimeter are usually generated by the application of short high voltage pulses between two electrodes for a minimum duration of micro to milliseconds. During treatments, the food material is in close contact with the electrodes and therefore is subject to an electric field. Assuming the sample homogeneous, the average electric field strength is as follows: E is equal to V by D, where V is the voltage across the food sample and D is the distance between the electrodes. Pulsed electric field has been applied in products such as milk and milk products, egg products, juice and other liquid food products. The benefit of pulsed electric field treatment is that it provides the pasteurization effect and results in better retention of nutritional and sensorial qualities than the conventional thermal pasteurization. The use of intense, short, high-frequency pulses induces slight heating of the product while the bacterial inactivation effect remains. This lack of heating will benefit the sensory quality of the product. During pulsed electric field treatment, the applied electric voltage transfers to the product and open up the cell and form pores. The pulsed electric currents are transferred to the sample placed between a set of electrodes inside a chamber, usually at room temperature. The distance between electrodes is termed as the treatment gap of the PEF chamber. Food sample is capable of transferring electricity due to the presence of several ions. When electric field is applied, the electric current flows into the food product and is transferred to each point in the same sample because of the presence of charged molecules. The electric field may be applied in the form of exponentially decaying square wave, bipolar or oscillatory pulses and at ambient, subambient and slightly above ambient temperatures. After the treatment, the food is packaged aseptically and stored under refrigeration. PEF technique is restricted to food products with no air bubbles and with low electrical conductivity. The maximum particle size in the liquid must be smaller than the gap of the treatment region in the chamber in order to ensure proper treatment. Regarding PEF processing system, A simple pulsed electric field setup consists of a high voltage pulse generator, a treatment chamber, and a control system for monitoring the process parameters. The high voltage pulse generator provides electrical pulses of desired voltage, shape, and duration. The treatment chamber consists of at least 2 electrodes with an insulating region in between where the treatment of the product takes place. One electrode is connected to the voltage source and another to the ground. The electrode material may be carbon, gold, platinum, metal oxide, carbon brass electrode, but the most suitable choice is stainless steel electrode. An insulating material holds the electrodes in fixed position and forms the chamber containing the foods to be processed. Insulator materials used are polythene, polypropylene, nylon, polysulfone, plexiglass, PVC, etc. A schematic diagram is shown above the pulsed electric field setup for your reference. The treatment chamber may be static or continuous. The design of the treatment chamber, example, the geometry and the dimension of electrode determines a more or less uniform distribution of the field inside the food. The more uniform is the electric field, the more homogeneous will be the treatment. The control system consists of 2 major devices, an oscilloscope and a temperature probe. The oscilloscope measures the voltage across the treatment chamber and shows the output voltage shape. Sample temperature is measured with the thermal probe placed inside the treatment chamber. Next, we'll see about the microbial inactivation by pulsed electric field. This can be explained by electromechanical instability theory. The cell membrane acts as a capacitor filled with dielectric material of low electrical conductance and dielectric constant. Accumulation of charges with opposite polarity on both sides of the membrane leads to naturally occurring perpendicular transmembrane potential of about 10 mV. When it is exposed to an external electric field, it charges along the electric field lines, resulting in the deformation of cell membrane. When the overall potential exceeds a critical value of about 1 volt, depending upon the compressibility, permittivity and initial thickness of the membrane, the electrocompressive force causes a local dielectric rupture of the membrane, inducing the formation of pore acting as a conductive channel. In simpler terms, when biological cells are exposed to electric field with high electric strength, the permeability of cell membrane is altered and the structural changes are induced due to the field open up the pores in the cell membrane. This mechanism is called as the electroporation. The permeabilization of a cell membrane happens in 2 key steps. Initial pore formation induced by the applied electric field, and next the formed pore has to be stable enough to allow interaction of the intra and extracellular media. A schematic representation of membrane permeabilization due to PEF has been given for your understanding. The changes are reversible if the pores are small in comparison to the membrane area. Increase in electric field strength and intensity of treatment will promote the formation of large pores in which the damage is irreversible. The irreversible breakdown is associated with mechanical destruction of the cell membrane and cell death. PEF treatment inactivates vegetative cells of bacteria, molds and yeast in various food products, but spores are not destroyed due to its tough protective coat. Yeast are more sensitive than bacteria. Also, gram-positive bacteria are more resistant to pulsed electric field than the gram-negative organisms. The applications of PEF mainly focus on foodborne pathogens and spoilage microorganisms, especially for acidic food products. Various factors that influence the microbial inactivations are intensity of electric field, pulsed wave shape, conductivity of the medium, pH, energy input, treatment temperature and time, type of microorganism and its growth stage. Induced transmembrane potential is directly related with the size of the cells. Yeast cells possess greater resistance than bacteria due to its bigger size of yeast cells. The sensitivity also depends on the growth stage of the microorganisms. The organisms are more susceptible during log phase, whereas they are resistant in stationary phase. A decrease in pH and increase in temperature have a synergistic effect on the microbial inactivation during PEF treatment. Presence of fat globules, agglomeration of microorganisms and presence of air bubble in the product are few other factors that affect the microbial inactivation. For the inactivation of enzymes by pulsed electric field, it requires a stronger electric field than microbial inactivation. Pulsed electric treatment at 21.5 kV per centimeter and high energy input reduced the lipase and peroxidase activity of raw milk by 65% and 25% respectively, with negligible effect on alkaline phosphatase activity. It has been reported that the effect of PEF on sensitivity of enzymes vary from type to type. On application of PEF treatment, a significant reduction of 70 to 85% in the activity of lipase, amylase and glucose oxidase are observed, whereas at the same treatment condition, peroxidase and polyphenol oxidase show a moderate reduction in activity of 30 to 40% and alkaline phosphatase showed only 5% reduction in its activity. It has been suggested that the inactivation of enzymes by PEF is actually caused by thermal effects. A complete inactivation of peroxidase was observed in PEF treated watermelon juice than heat treated juice. A study on the PEF treatment of papain showed irreversible reduction of enzyme activity. The papain activity was not recovered after 24-hour storage at 4 degrees Celsius. The reduction in activity of papain is due to the loss of alpha helix structure in papain molecules and also reported that the heat contributes mainly to the observed inactivation effect. Regarding the effect of PEF on food quality, orange juice was PEF treated and compared with the thermal pasteurization at 94.6 degrees Celsius for 30 seconds. PEF treated juice retained higher amount of vitamin C and flavor components than conventionally pasteurized sample. In case of flavor components such as decanol was found to be reduced by 40% during thermal pasteurization, while no such loss was observed by PEF treatment at 30 kV per centimeter, either at 240 or 480 microsecond. It has been reported that even if some of the components suffered losses during PEF treatment, it will be always in less proportions than the heat pasteurized juice. Such effects were also observed in case of PEF treated apple juice. PEF treated orange carrot juice was compared with the heat treated juice. The shelf life and ascorbic acid degradation rate of both the samples were investigated, and it was observed that the carrot juice was 83%, whereas in the PEF orange juice it was 90%. PEF treated orange carrot juice extended the half-life of the juice stored at 2 degrees Celsius to 50 days compared to the heat treated juice. High intensity PEF treated watermelon juice maintained a brighter red color than the heat treated juices throughout the storage time. Let me see about the applications of PEF. The first is the pasteurization. PEF technique is used to pasteurize a variety of liquid and semi-solid foods such as milk, yogurt, apple juice, grape juice, orange juice, sugarcane, soups, brine and liquid eggs. This technique can also be coupled with other processing methods. It is highly suitable for heat-sensitive juices and soup in order to prevent the loss of vitamins. It has been investigated that after PEF treatment, the nutritional quality of milk remained unchanged with no loss of vitamin B1 and B2, vitamin D and E, but vitamin C showed 93% retention. During the PEF treatment of sour cherry juice, significant reduction in the inactivation of E. coli, Staphylococcus, Listeria monocytogenes, Erwinia catorrhea, Pseudomonas and Botrytis and Penicillium were noted, with increase in the electric field strength and treatment time. In 1996, Food and Drug Administration approved the use of PEF in the preservation of liquid eggs, while the first commercial PEF application for fruit juice preservation was installed in US in 2005. The alternative applications of PEF include enzyme activity modification. Second application is drying. Apart from the microbial inactivation, due to the opening effect of cells, the technology can also be used to facilitate rapid removal of moisture from the cell matrix. Thus, it finds its application to enhance the chemical reaction and drying process. The principle of permeabilization due to PEF is successfully used to enhance mass and heat transfer to assist drying of plant tissues. Such results were observed in apple, mango, and potato. PEF technique has been applied as a pre-treatment before osmotic dehydration of carrot. The samples were exposed to different levels of exponential pulses with a total specific energy input in the range of 0.04 to 2.25 kJ per kg. The electric field strength was in the range between 0.22 to 116 kV per centimeter and pulse duration between 378 and 405 microsecond. The PEF carrot samples were further treated with sucrose solution at 40 degrees Celsius for 5 hours and the results were compared with the untreated osmotically dehydrated sample. It was observed that PEF treated samples showed a reduced moisture content and increased solid content during osmotic dehydration. The third application is extraction. PEF is also utilized to enhance extraction of sugars and other cellular components from plant cells such as sugar beets. The application of PEF with a peak voltage of 20 kV has been used for the treatment of fruit mashes. Avoiding an enzymatic maceration due to PEF retains the pectin in native and highly esterified structure. This provides a potential to extract high quality pectin from the pomace after juice extraction and therefore a step toward more economic and sustainable processing. During oil extraction process, by both pressing and solvent extraction, the application of PEF as a pre-treatment increased the oil yield. The PEF pre-treated oil seeds also showed a higher concentrations of tocopherols, polyphenols and polysterols after extraction. Higher juice yield of about 75% were observed in PEF treated wine grapes compared to the control sample. Also, PEF treatment increased the total anthocyanin content in grapes. Almost 3 times higher anthocyanin content was reported in PEF treated grapes than that of the untreated grapes. PEF also find application in reducing the solid volume or sludge of wastewater. Due to the breaking of cell walls during PEF treatment, the cell contents are released to the surrounding. Those burst cells are much more susceptible to other biological and chemical processes such as anaerobic digestion, which can significantly reduce the solid content or sludge in wastewater treatment facilities. Advantages of PEF includes that the process almost maintains the product quality without thermal and mechanical stress. It does not degrade the nutrient properties. The process does not produce any harmful byproducts. And the setup has no wearing parts. PEF treatment increases the extraction yield and it is able to process pumpable food products. PEF allows structural modification of product. For example, tough and inconsistent produce such as sweet potato, turnip and beetroot become easily processable with PEF. PEF as a treatment before drying helps in softening vegetable tissues and improves the mass transport of water. It is also used to improve the peeling efficiency due to softening of tissues. It is considered as a cost-effective process because it uses the latest advances in the pulsed power technology. This technique can be integrated into the production lines. No labeling in contrast to radiation and heat sterilization. Highly suitable for extraction of secondary metabolites which are unstable at temperature and leads to mechanical stress. There are some limitations too. PEF techniques are suitable only for liquid foods and paste regarding to the pasteurization. It needs to optimize the process on solid matrix including through the design of new contact cells regarding to the permeabilization pre-extraction. High cost of initial investment and high energy dissipation. We have now come to the end of this module. Let me summarize the top points. PEF is one of the promising non-thermal techniques to inactivate microorganisms. It provides the pasteurization effect and results in better retention of nutritional and sensorial qualities. PEF setup consists of a high voltage pulse generator, treatment chamber, control system for monitoring the process parameters. Electroporation is the principle behind the microbial inactivation by PEF. PEF inactivates vegetative cells of bacteria, molds and yeast in various foods, but spores are not destroyed due to its tough texture. Various factors that influence the microbial inactivation are intensity of electric field, pulse wave shape, conductivity of the medium, pH, energy input, treatment temperature and time, type of microorganism and its growth stage. Stronger electrical field is required to inactivate enzymes than microbial inactivation. PEF finds applications in drying, extraction of oil, bioactive components, pasteurization, enzyme modification, tissue softening and sludge treatment. PEF treatment samples retain the nutritional and sensorial characteristics compared to heat-treated juices. Hope you have got an insight about pulsed electric field and its applications in food processing. Thank you for your patience hearing. Happy learning. Thank you.